Hi all, how well do you know about culture media and culture techniques? Test your microbiology knowledge with this quiz on culture media and culture methods. So this quiz covers types of culture media and culture methods for isolating and identifying microorganisms. Like, share and subscribe for more microbiology quizzes and tutorials, and turn on notifications to stay updated. So the first question is, agar concentration required to prepare nutrient agar is. The options are 2%, 6%, 0.25% or 0.5%. Agar concentration required to prepare nutrient agar. So the answer is 2%. So to prepare a solid culture media, the concentration required is 2% of agar. And if it is 0.5%, it becomes semi-solid. And as you know, liquid culture media does not need any agar, so it is 0%. The next question is, which is an enriched medium of the following options? A. Selenite F broth, peptone broth, McConkey agar or chocolate agar. Which is an enriched medium? The answer is chocolate agar, because enriched medium is a media which has additional nutrients in it, like blood or serum or egg. So this chocolate agar contain blood. So it is the enriched medium. This one, selenite F broth, it is not an enriched media. It is used for isolation of salmonella from feces sample. And peptone water, it is used for growth of microorganisms in laboratories. This one also is not an enriched media. And McConkey media or McConkey agar, it is used for differentiation of bacteria which ferment lactose and which does not ferment lactose. So it is a differential media. The next question is, recommended transport medium for stool specimen suspected to contain Vibrio cholerae. Transport medium for transportation of stool specimen which is suspected to contain Vibrio cholerae. The options are selenite F broth, Venkatraman Ramakrishnan medium, nutrient broth or blood agar. The answer is Venkatraman Ramakrishnan medium. Carrie Blair medium also is a transport media used for Vibrio cholerae. Next question is, Robertson's cooked meat broth is an example of. Options are enriched media, enrichment media, anaerobic media or solid media. Robertson's cooked meat broth. The answer is, it is of course an anaerobic media which does not contain oxygen, which is used for cultivation of anaerobic microorganisms. Thioglycolate medium is another example of anaerobic media. Robertson's cooked meat broth is anaerobic. Next question, blood culture bottle contains A. Peptone water broth, BHI broth, selenite F broth or tryptic soy broth. The answer is BHI broth or brain heart infusion broth, which is used for in the blood culture bottles. Next question is, Lowenstein-Jensen medium is used for isolation of which of these organisms? Lowenstein-Jensen medium is used for isolation of options Mycobacterium tuberculosis, Vibrio cholerae, Salmonella or Shigella. Of course, Lowenstein-Jensen medium is used for cultivation of Mycobacterium tuberculosis. Next question, thiosulfate citrate bile salt sucrose, that is TCBS agar, is used for. Options are A. Vibrio species, Salmonella, Shigella or E. coli. thiosulfate, citrate, bile salt, sucrose, TCBS agar? The answer is, it is used for the growth of Vibrio species. Next question is, which is not a selective medium? Out of the options, deoxycholate citrate agar, xylose lysine deoxycholate agar, selenite F medium, or Wilson Blair bismuth sulfide medium, which is not a selective medium? Is it DCA, that is deoxycholate citrate agar, XLD, that is xylose lysine deoxycholate agar, selenite F medium, or Wilson Blair bismuth sulfide medium? The answer is selenite F medium, because selenite F medium is a broth. Broth means it is a liquid medium. So selective medium is always a solid media. It is an agar based solid media. So selenite F broth is a liquid media. So it is not a selective media. It is an enrichment media. So it is selenite F medium is the answer. Next question. Which culture method is used for carrying out antimicrobial susceptibility testing? By Kirby-Boer method. Which culture method is used for carrying out antimicrobial susceptibility testing? Is it streak culture, lawn culture, stalk culture, or stab culture? The answer is lawn culture. Lawn culture is used for obtaining uniform growth of microorganism, which is used in the case of antibiotic susceptibility testing. Whereas streak culture, it is used for isolating a pure strain of bacteria from a mixed population. So it is streak plate method. It is used for isolation, streak culture. And stalk culture, it is culturing the organism as a zigzag line. So it is used for pure bacterial cultures. In tubes, in test tubes, we use stalk culture. It is done on the agar slopes or slants. It is called as stalk culture. And stab culture, it is again a method of growing bacteria in a test tube using solid agar, in which it is inoculated by stabbing using a straight wire, not a wire loop. It is done by using a straight wire. So these are some questions on culture media and culture methods. Thank you.